a monohybrid and dihybrid cross. And I'm going to fix this. This is being a little goofy again. And that's okay. So with this monohybrid and dihybrid cross, we have the idea that we could have, for instance, two different genes involving two different traits. In the case of the example I gave you, we had flower color controlled by gene A and plant size controlled by gene B. So that's totally different traits. And we had two different versions, two different, what? What are these versions called? Alleles. Two different alleles for each of those traits. So we had something where we had basically a purple version and a white version, but the purple is dominant over white. And we have a tall versus a dwarf version, with the tall being dominant over the dwarf one. So we had this dihybrid plant, and it's able to produce gametes. When it does that, when it does meiosis, one of these chromosomes and one of these chromosomes gets into each gamete. It needs to be one of each. If it's two of those and neither one of these, that means the gamete is missing an enormous amount of information the plant has to have to survive. It'd be the same with us. You wouldn't just be missing this gene, you'd be missing hundreds or thousands of others. So you must have one of each of these chromosomes. Now, I had a question along this line on the test, and most people understood it, and not everyone did. This is something that fools a lot of people, so keep that in mind. Meiosis isn't just grabbing, say, if you're cutting the number of chromosomes from 4 down to 2, it's not just 2 at random, exactly. It's one of these plus one of those. So one of those can be at random, and one of those can be at random, but it has to be one of each. It can't just be those two or those two. That would not be a viable gamete. If that happened in your case, you'd be producing, say, an egg that's missing information on how to make, say, stomach enzymes and blood types and proteins for your eyes and so on, and that gamete would not be a viable one. The resulting fetus would probably spontaneously abort. It would be a miscarriage. So you end up getting one of these chromosomes plus one of those, producing these 4 different gametes, which then we can look at the mating of the two hybrids with each other, producing this complex Punnett square. And that was something where we had then 9 out of the 16 outcomes had the dominant characteristics. But only one of them was true breeding, big, 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 big. All the rest were hybrids to some extent. And then we had 3 versions out of the 16 that were dominant on one trait and recessive on the other. And then 3 more where you switched that idea around. And finally just one that was recessive. Looking at the phenotypes. So that's what happened with the dihybrid cross. A little later we're going to see a variation of this. This diagram will sort of come back, but it's going to involve dogs, and it will be a little bit different now. But that is where we actually left off with this stuff last week. Now, I don't want you to think that when we're looking at genes, the only option is that a gene has 2 alleles and they're either dominant or recessive. The reality is often much more complex than that. So you can have, for instance, incomplete dominance. In this case, if you take a snapdragon that's red and you mate it with a white one, you end up producing hybrids that make pink flowers. This is incomplete dominance. What happens is the hybrid has one of those genes and one of those. And when you combine those, you don't get as strong a red effect as you would get in this case. Instead, it's watered down. It is pink. The red pigment is basically diluted. And it looks like we've actually done blending inheritance. If we stopped right there, we might say that, oh, these genes got smooshed together and now we've got an average set of genes going on. But that's not true. if we go and mate two hybrids with each other, we get both extreme types coming back. So how do we get this extreme type coming back? How do we get that red one coming from mating two hybrids? How did this happen? Yeah, each of these had a big R and little r. Well, the two big R's landed there, the two little r's landed there, and then here again, you got one of each. So when you go to another mating generation, it shows us that these versions of the genes came through intact. The versions of the genes didn't get changed. So this is something where you can have incomplete dominance, where you don't get a full dominant behavior. If it was full dominance, this would look exactly like that one, but instead, it's not that way. So a lot of the things that go on in us really work this way. You could be looking at, say, something involving normal skin coloration, not an albinism, but otherwise normal genes involving normal gradations in skin color. They're going to work more this way, more along the line of incomplete dominance. So I've got text there we don't really need. Blood is something, though, that I wanted to get into. So we look at red blood cells. Well, can you just stick any red blood cells into any person? I see some head shaking. Why? What could go wrong?
You have different blood types.
Yeah, and they may be incompatible. Well, what happens is all of your cells are covered with proteins that are identifying those cells to your immune system. Your immune system includes a vast number of proteins, defensive proteins, that are circulating through your plasma, through your blood, through all of the different spaces in your body. And these defensive proteins, these antibodies, or immunoglobulins, as they're known, those can bind to foreign molecules, and they'll stick to them and mark them for destruction by your immune system. So what you have are lots and lots of proteins that are on your cells, and they're identifying your cells to your immune system, saying, don't attack me, I'm on your side. And usually, things don't go wrong. Now, people may have autoimmune disorders where their immune system does start attacking their tissues, but most people don't. And so your cells will have these markers on them, what we call antigens, that are identifying them and saying essentially which team they're on, which body they belong in, and then your immune system can come along and have its antibodies bind to things that don't belong there. So if you do a transfusion and it's incompatible blood, let's say a person has severe blood loss, is in the emergency room, he's type O. They give him type A blood. It would trigger an immune response where the immune system would try to kill that new blood, probably end up killing the recipient in the process. That would be an example of an incompatible transfusion. Let's just see some of what goes on with this. I'm going to raise the screen and do some markings up here on the board on all of this. Then we can deal with an emergency room situation. So I'm going to pass out to you an exercise here, and this is what we'll be working on through. This will be worth 3 points, so I want to get it today. So, here's, let's not do that. So here's what happens. In the case of the blood types, what are the common ones that we talk about?
A.
Sure.
B.
B.
O.
O.
AB.
AB. There's something else we talk about regarding blood types.
Positive and negative.
Yes. All of these can have positive or negatives. Now there is one gene called the ABO gene that controls all of this. That controls what we call the blood type letters. You'll also have what's known as the RH gene. And that controls the positive or negative part. So really it's two different genes together, giving us a total of 8 blood types that we would normally consider. Now, right now we're only going to be looking at the ABO gene, which gives us 4 different possible blood types. And it's based off having 3 alleles for this blood type gene. So you have the ABO gene. That can have actually 3 different versions. One of them, you write it this way, capital I with a superscript A. That together is a description of one allele. Which of these do you think that might be tied with?
A.
How about A? Also AB. The second version of this is capital I superscript B. Connected with that one, and again with AB. Or little I. That is connected with type O. So instead of just having one gene that's either dominant or recessive, which is how we started off looking at this, we've got more complexity now. We have actually 3 versions of the gene, giving us 4 different versions of the phenotype. And in addition, there's a second gene creating some variation on that. So now we're starting to look more at the reality of how complex genetics can get to be, at least more complex than just looking at the pea plants with the flowers. But the flower part was essential in understanding how this works in the first place. Just don't think that that's the end of the story when it comes to how complicated genetics can be. This is a good practical example of involving actual medicine, involving a Nobel Prize that was given out in 1920 for scientist Landsteiner, who came up with all of this. And this has saved millions of lives since then. It's a vital thing in medicine. Anything that they're trying to do an organ transplant, obviously they have to get this kind of thing straight, never mind a blood transfusion. So this is critical in medicine and certainly deserving of that Nobel Prize. So, what's going on here? All of these are the phenotypes. If we're talking about somebody being, type A negative, that's a phenotype. It's basically looking at how the blood responds if you challenge it with different antibodies. You'll see whether or not the blood clumps up in little globs instead of staying smoothly spread out in a dish. If you take some mystery blood, put in antibodies like an anti-A serum or an anti-RH serum, an anti-B serum, then you're able to see how the blood behaves. And this is what can be quickly done, say, in an ER. Somebody comes in and they're desperately in need of blood. They're not just going to immediately stick blood in you. They're going to check and find out what your type is. And it only takes about 5 minutes to do. So they don't have to go sequencing your genes. All they have to do is look at how the blood behaves. When you put in, have a little bit of blood, like a couple of drops, and you put in a drop of anti-A serum, you'll find out quickly if they have this particular gene, version of the gene. Another test and another test, and you've got the blood typed. It's very quick and easy to do. So all of that is the phenotype. The underlying genotypes are actually different combinations of these. Everybody is going to get some of these. Would anybody get all 3 of them? Well, you've got a chromosome. If you're looking at your pairs of chromosomes, I forget which one it is. Let's say it's chromosome number 7. How many chromosome 7s do you get? 2. And those chromosome 7s could have 2 identical versions of these or 2 different ones. They're not going to have all 3. So any given person is getting some combination of 2 of these. That'll be 2 that are the same or 2 that are different. They'll either be homozygous or heterozygous. And that's true with our genes in general. You're either homozygous or heterozygous with the different genes that you have. So that means there's more variability out in the population than there can possibly be inside of one person. That's also going to typically be true. So imagine we've got a person who is type O, or type A. I'll start with type A. They have their red blood cells. The red blood cells have these antigens on them. What these antigens are, they are proteins that are embedded in the cell membrane, and then they have these little groups of carbohydrates attached to them, about maybe 10 or so sugar monomers that are put onto them in a particular arrangement, so they become what we call glycoproteins. Always a protein that's embedded in the plasma membrane, and then with this little carbohydrate tag that is extending out past the cell, and that is marking what kind of an antigen it is. So let's say the type A ones look like this. They don't, but let's imagine they do. These are going to be the markers that are saying this is type A blood. Now, if you already have type A blood, you have this in your bloodstream, and your immune system knows to leave it alone. If we take type A blood from someone else and put it in you, the immune system will just treat it as being more of you. That'll be okay. That's compatible. But if you put this into someone with type B blood, they're not going to have an immune system that's trained to think that's okay. Their immune system will encounter these, and it will think, oh no, there's a bacterial invasion going on. I have to destroy those bacteria. And it'll go to work on destroying these cells. Not what you want if you're getting a blood transfusion. What sort of genotype could possibly give us that? Some combination of 2 of these. IA, IA. So the genotype for that can be this. That will give us what I just drew. If we have instead someone who's type B, they again will have antigens, but they're going to be shaped a little bit differently, maybe like this. What could be the genotype for that person based on this? IB. As we'll see in a second, that's not the only way to get a type B person. But that would be someone who's homozygous B. They would be like that. Their immune system is used to having these around. It leaves them alone. It will not leave these alone. How about an 80 person? What will happen in their case? We're going to have both of these. They'll be heterozygous, one of each of these dominant types. And what that's going to do is make half of them like this and half like that. Sorry. So it's a mix. That's the AD person. Last one, type O. Little I. And what happens there, with the glycoproteins, you have the protein part, but you don't have the carbohydrate tag. So these are truncated antigens. They're missing some of the material that shows up on the others. These do not trigger an immune attack. Everyone's immune system just leaves these alone. So this is the safe kind to have in the sense of this being something that could be put into anyone. Essentially, anyone's immune system would say that this is okay. Now we still have 2 genotypes though to worry about. Is there another way that you could get the type A response? Remember that when we're talking about the type, we're talking about how the blood responds. If we go and mix it with an anti-A, say, serum, what are you starting to say? IA little I? That's right. If it's IA little I, then this is what happens. Every other one of these is missing the carbohydrate tag. This blood will still act like type A blood. If you mix it with an anti-A serum, the anti-A antibodies will attach to these, and that's enough to cause the same kind of reaction. And likewise here. So that's the other way to get a type B as well. So we have these 6 different genotypes, and that's what I just marked along the bottom here. That's simply looking at the ABO gene. The positive and negative is referring to another one called the RH gene. That's a different marker, and there's only 2 versions with that. You either are RH positive, where you are creating that marker, or else RH negative, where you're not. The way it works with the positive and negative is that a positive person can receive positive or negative blood because they already make the marker. They're fine if donated blood has the marker or if it doesn't. But a negative person doesn't make that marker, so the negative person has to get RH negative blood. Positive can take anything, negative just has to get negative, with then compatibility on the other gene applying as well. So given all that, I'd like you guys to work on that worksheet. And notice the advice column letter that's on the back there. That was an interesting one that I read a few years ago. I like these online advice columns, believe it or not. And there was one that seemed classically perfect for our class material, so I held on to it. See what advice you would give that person in this case. Does everybody have a copy? Okay. Did you get 2? Is that 3? Okay, so let's see what we get with this. And if you want, maybe now would be a good time to come down and pick up the quizzes. Okay, yes. When I do that in the future, you should be all this response and just say, how do you respond to this? That's a special just say, it looks to me like our yes.
Oh, actually,
For these.
Now, this table that's on the front here, I'm going to put up one little hint here with that table. 9 letter A's, 7 letter R's. So there's 7 ways the blood gets rejected. Pay attention to who is donating or who is receiving the blood and who is giving the blood there. People sometimes mix that up. But that's on the first page. Second page has to do with paternity issues. That's a question about who's the daddy. Now, ever since we've had the blood typing information available, we've been able to use it to try to help settle paternity cases. That usually comes up if, say, a woman gets pregnant and the guy hasn't married her and she is saying, hey, I need child support. That guy, I was going out with him for a while, he got me pregnant, and then he dumped me and took off. As soon as I was pregnant, he took off. He owes me for 18 years of child support. And a judge would look at that and have to decide whether or not the guy probably is the father. This information is helpful. When you're looking at those paternity issues, assume that we know who the mother is. That would be hard to get mixed up. So assume that we know who the baby is, what the baby's blood type is. We know who the mother is and what the mother's blood type is. Therefore, we know what sorts of alleles are in each of those people. Look then at the accused father and see whether he could have provided the other information that the baby has, the other allele that the baby has. You may decide that in the end you can't be sure, but in some cases you can make a definite decision. Then the third part of that third page, that's basically imagining you were giving the advice. What advice would you give to the person who wrote in there? We'll be right back.
Oh, dang it. I thought it was like mixing them. I was like, wait.
Even if it was like that,
Yeah. Oh, recipient. Oh, different gender. This is all, and then rejected. This is rejected. And then B accepted. Rejected. It's accepted, right?
really? Yeah, maybe is
I'm doing it for Ivy. So I don't have to like show her the thing later. Yeah. Doing this. Yeah, this means accepted, and then these are rejected. Okay, next one.
this is universal recipient. So A, B can accept everyone. Universal donor is
Oh.
O, because O will be accepted by the person right here, and then human
No, this would be wrong. Cause you remember in Auto Concerto, like, right?
Yeah, but the baby could only have II. That's the only one. Like, for what?
The mom is type A.
So she can have A,
IA.
no.
Or, oh, sorry. Cause up here. That one.
Oh,
The dad?
no. He can't be the dad. The dad's just maybe. Yeah, I know. I get confused because they're all.
It's so weird.
Like, it's A.
Because of this.
It's no. Because some baby would somehow have to have an IO from someone else. And not someone else is not the dad who's being accused of being the dad.
He's B.
No, because in Rencorn, she did not give. Rencorn, she was type B, and he couldn't give it to what's his name, Wu Chen's daughter.
The baby's type is B. Oh. Shit, that scared me. Okay. And then the man.
Yeah, I think he's.
The man is II, right?
Should he be? No? No? Because he doesn't have an IB allele to give. But the mom.
No, but the mom has a B because then she can give one of this and then one of that.
Oh.
Let me ask him.
No, I think you're right.
Let me ask. Just to be sure. Mr. Cooper, for this one,
Yes.
would it be yes because like the mom can pass this because like the dad?
Yes, yes, all you're saying is he could be the father?
Yeah.
Okay, sure. If the baby is that and the mother gave an IB, this guy could have given a little I.
Okay.
You're not proving that the guy is the
Yeah.
father, but it's possible.
Okay.
And in this case, you prove he can't be.
Yeah. So it's right.
I just said this one is yes because like the earlier one, you have one allele from the dad and like whole.
Wait.
No, it's not possible. What am I saying? It's not possible.
This is kind of interesting. We're doing forensics. Like, no, it's not possible.
Yeah.
That's not the baby. It's not.
Yeah.
The dad is.
I was looking
The
at
dad
it.
is correct. It's not the
I was
baby.
looking at it and I was like,
Yeah.
no, I was like, no, what if it's not mom's baby? What do we do then?
Okay, wait. Let me take pictures of these for her. Oh my god. Okay, all you have to do is an analysis, so I'm gonna end the video.